And I think I hope that you can understand that from the workshop that we've had and the lectures from today, the importance of the objective. So it's sorry, I realized that my phone was not plugged in, so maybe you couldn't hear me. So I hope that you realized from today's lectures and the workshops that the objective is very important. It's very important that you choose the correct objective and you treat your objective well, and that you design your sample in the correct way so that it matches with your objective. So now there's one thing that is also overlooked very often in trainings, unfortunately, is how do we find the area of interest, and also how do we focus? So this is what we're going to talk about now. So tiling is a way to increase the focus. It's the field of view. It's very useful. So if we have one objective that we have chosen for reason X, Y, Z, and we have a certain field of view, but our sample is bigger, then we can take many images and then stitch them together in such a way that in such a way that we artificially increase the field of view. And this is a very useful technique that can be used for live cells in here, but can also be used for fixed samples. So you can see here, this sample is very big. This is 1 millimeter, the scale bar. And what we have is that we have a small region of interest that we need to look for. And the last thing that you want to do is bleach your sample before you even image it, because, you know, we are spending a lot of time preparing our sample and making mice and all these sort of things. And why do we start destroying it even before we acquire images? So what you want to do instead is to use the automation of your microscope, if you're lucky enough to get an automated microscope, because if you do not have an automated microscope and you have a manual stage, then you cannot do what I'm going to talk about now. But then clearly, I would love to convince you to change microscope and get a microscope with an automated stage. When you get an automated stage, you really are able to enhance completely the type of experiments that you can do. So when you have this automated microscope, you can make like a big tile that covers the whole sample. And again, this is called different things. There's mosaic and large image and overview and all sorts of stuff, but it's the same thing. So it's a stitched image, a big image that is stitched made of many tiles. And the way we train our users at our facility is that they find the sample without looking in the eyepiece to start with. We're looking in the eyepiece, but not at fluorescence, looking at transmitted light, so they don't bleach it, just looking for the sample. And then afterwards, we make a big overview, and we use this overview to navigate in the sample. So in all microscopes that have an automated stage, once you have the overview, you have the possibility to move using the image. You can move in the image. And for example, in the Nikon software, you can right-click in the image in the place where you would like to go to, and then you right-click and you say move to this position, and then the stage moves to the position that you have shown in the image. In the Zeiss software, you can do exactly the same thing, where once you have opened the big overview image, at the bottom of the image, you have something called stage. And if you click, once you have clicked, you have activated the stage button. If you click in your big image, the stage will move to that position. So this is, this, I'm sure it's the same with Olympus and Leica. I just don't know what these functions are called, but it's the same thing. So it is really important to learn how to use this, because then you can make an overview image and then navigate in your sample using the image instead of continuously bleaching the sample. Another thing that is very useful is that if you have, okay, here we have only 2 regions of interest, for example, but if you have more regions of interest, if you look in the eyepiece and you try to find them, it's quite easy for you to actually image twice the same. So when you have the image, it's much harder to make such a mistake, and it's easier to see where you have imaged already. So this is a very convenient way to image. And when you come to us for the your student imaging challenge workshop. So when I image your sample, I'm going to show you how convenient it is to take an overview image and use it to navigate in your sample. So this is a very good way to find the area of interest in a large sample with minimum bleaching. So you use a low magnification when you want to do the overview image. You're going to use a low magnification objective. And then you use, for example, the minimum channel that allows you to find your region of interest. So for example, in here, we also acquired the blue image, but we didn't have to. I think if we had only the red channel, it would have been enough to find our region of interest. It depends on your sample, basically. And then you do not use any averaging, and you use you save the reference image, and you can use this also to show the context of what you have imaged afterwards when you are imaging single regions. So this is also very powerful to use when you are publishing. And you can use the full field of view, so no crop, no zoom, so that you have the field of view as big as possible. And if you're using a single point confocal, you should use large pixels, so you're undersampling. We will see what undersampling means next week, but you will use large pixels, so it goes much faster to image this very big overview image. You're going to use a big open pinhole. This way you will be able to lower the laser light and not expose your sample so much while you are taking the overview image. You can use the bidirectional scan. It's the same thing. Many people who use a single point confocal do not know that there is a monodirectional and a bidirectional scan. And with the bidirectional scan clearer, you go twice the speed. So it's very convenient. If you have a resonance scanner, this is the type of things that are perfect to acquire an overview image. And then a short pixel dwell time. And if you use a camera-based system, you should use a low illumination and bin the camera. So when you bin the camera, the image becomes brighter, and so you can reduce the illumination. So we will talk about binning the camera and what that means next week. But basically, this is a very powerful technique to find your area of interest and navigate in your sample with minimum bleaching. So I recommend that you adopt this technique. And so once you have found the positions, you can then go back to these positions, as I said, by just moving the stage using the image. And then you can, for example, acquire Z-stack there, or with all the colors that you need and the correct pixel size, and maybe doing some averaging to reduce the noise, etc. All systems that are automated in XY can do this. It doesn't matter which company they are from. So please, if you have an automated stage, use this technique to find the region of interest. So another way to have an efficient exploration of the sample and have an overview plus re-imaging is to include automated image analysis in the pipeline. So many systems nowadays can do this sort of things, but it requires that your microscope is able to have a pipeline, allow you to make a pipeline. And so most manufacturer software sell at least a plugin that allows you to do this type of thing. Sometimes it's an additional piece of software, so maybe you need to buy it, but maybe you have it on your microscope. So I think you should figure out if you have it or not. So in Nikon, it's called Jobs. It allows us to design the pipeline the way we want. And I think in Zeiss, it's called, I forgot, it's called flexible developmental something designer, something like this. Experimental designer, something like this. And so basically, we're going to get images of our sample. These are yeast images. And we take an image of our sample that is looks really crap. Okay, fine, doesn't matter, because it allows us to see anyway what we're interested in. And it allows us to see that, for example, if I'm interested in a pink thing with a green thing next to it, then I know I do not want to image this and I do not want to image that. Okay, so I'm going to only focus on those that have pink and green next to it. And so I acquire an overview image of my sample that is very low, noisy, with low resolution, and then I put in my pipeline an automated image analysis that is going to find these areas of interest. And then the software will log the XY positions. And after that, we can do some re-imaging, which is automated, and you can go and get a coffee, and you can see here it's much higher resolution, and I only have the positions of interest in these images, so I can collect thousands and thousands of cells with only the area that I'm interested in, so I can go and do something else in the meantime because it's automated. Isn't that nice? Okay, so this is the proper way and the efficient way to navigate in your sample and explore and find the area of interest and re-image it. Another aspect of finding the area of interest is in Z, and so it's the focus. And that is a big issue because first we need to find the focus, then we need to make sure that the focus stays at the same focus so that we have consistency. And this is extremely important for image analysis, as you have seen, when things are, if there's a slight refraction index mismatch in any way, and clearly it's never perfect, even if we put our efforts into it, then as soon as you're a bit out of focus and away from the cover slip, you're going to have haze, and then it becomes blurry and much harder to analyze. So in that case, we can try and use autofocus. So I'm going to tell you now a little bit more about focus. So the problems, typical problems that we can have with focus can be that on one side, if you have a long sample like this one, which is a piece of skin, on one side of the sample, you're focused, and then you take a large, like you take an overview image, and when you arrive at the other side of the sample, it's not focused, okay? So that is difficult. It's pretty much impossible to analyze an image like this. Of course, you can do a Z stack to try to compensate, but it takes much longer, especially on a single point confocal. So it's not ideal. Plus, it bleaches the sample, of course, at every Z level. So that is not ideal. Another problem that we can have is that even within one field of view, we have a difference in focus. And you can see in here that we have a difference in focus from the top left corner compared to the right bottom corner. And that is very, very difficult to deal with. So again, it will create some problems with image analysis, and you have no reliable intensities because this will look dimmer than this completely artificially. And so what we need is to have a focus strategy and a strategy with the start focus and a strategy to keep the focus. So this is what I call the focus strategy. So we can use autofocus functions, and these are extremely useful because then you don't need to bother. It will find the focus itself. But it doesn't work in all cases, but it works in many cases. So what are the applications for autofocus? It can be that you're doing a time lapse, so you're doing imaging over time, and there's always fluctuation in temperature and stuff like this. And so even if you are careful, there's always fluctuation in temperature, and therefore there will be a fluctuation in focus over time. So when you do a time lapse, you definitely must have an autofocus strategy. And then it can be also that you took your sample from the fridge and it's cold, and then you put it and you want to image it with an oil objective. And when you do that, all the temperature or the energy that comes from the energy transfer goes through the oil. And so that means that you are completely increasing the fact that using an oil objective, you are increasing the problem of the fact that the glass is changing temperature. So even if you equilibrate your cover slips for 1 hour, you will, with an oil objective, any changes in temperature will go through the oil, and so you will see an effect. So it is really difficult with oil objectives to actually keep the focus for quite a while. If you bring your sample for the cold and you put it on an oil objective, you will see the drift happen within seconds. Other reasons why we need a autofocus is when we are imaging over long distances. This is what I was showing you with this piece of skin. We have quite a few field of view, and then actually on one side of the sample, or if you have a multi-well plate from one well to another, you have a different focus. So that's another application. And yet another one could be if you are looking at something that is at a surface that is not flat. For example, if you have cells on top of a gel and the gel is never completely flat. So then this is also one application. So the major cause of loss of focus are the temperature drift, and also that the sample is not flat. It can be because the sample has not been mounted properly. So if you mount your sample with a hardening mounting medium, you need to leave, and even if you put it with a soft mounting medium and nail polish, we'll see this next week with the sample preparation lecture, you need to allow the sample to dry or the nail polish to dry when the sample is flat and horizontal. So do not mount your samples and then put them vertical in a box. Because the cover slip, the mounting medium will go down and the cover slip will not be straight, okay? So dry your samples that you have mounted horizontally so that we don't have slanted cover slips. Something else that can happen is that the sample doesn't sit flat on the stage. And this is a typical mistake of people wanting to put the cover slip flush at the end of the slide. So Gabby will talk about all this next week. But basically, we need to have a little space at the end of the slide where there's no cover slip. And she will explain to you why. So when the sample doesn't sit flat on the stage, we have a major problem with the focus. And of course, the bottom of multi-well plates is often not flat, and this is a pity. This is even worse if you put your multi-well plate in the fridge, in incubator, the plastic distorts, and then the bottom is not flat. It's a lot worse with plastic bottoms compared to glass bottoms, but it also happens with glass bottoms. So these are the major problems that we have with focus. So it is critical to have the sample always at the same focal plane for image analysis. It's very difficult for you to compare intensities because the intensity is going to change, the shape of the objects is going to change if the focus changes, and it changes very, very quickly. So to have reliable information for image analysis, you must fix the focus. So the solutions that we have to have no problem with focus is we have some autofocus solutions, hardware and software autofocus, and I will explain this now. And we can also, if we have a problem where we have within one field of view, as I was showing you in the previous slide, the left corner was blurry compared to the rest. So within one field of view, we have parts that are in focus and parts that are not in focus. The only way to deal with this is to make a smaller field of view and then some overview to increase the field of view. And then we can also, of course, add a small Z stack, but again, on single point confocal, it takes a lot of time. So let's have a look at hardware and software autofocus. Software autofocus is pretty much available on all microscopes that are automated in Z. And so what the software does is that it basically goes down a little bit. So you focus more or less on your stuff, and then it goes down a little bit. It's going to take some images and it will do a small Z stack. And so at some point, one of the images, so it images every time. At some point, one of the images is going to have a higher contrast. And then it keeps going above the sample, and then it keeps taking images. So this image is the one that has the higher contrast. So there is image analysis going on in the background that determines the plane where we have the highest contrast. And this is what the software decides. This is the best plane. So this is a very powerful technique. It works best with low numerical aperture objectives, which are not very influenced by all aberrations, for example, the refraction index mismatch. So this works best with low numerical aperture objectives. The disadvantage is that we're imaging the sample and therefore we're bleaching it. Then some of these are very fast, but it anyway takes time. So if you are, if you're imaging lots of samples, for example, it will really increase, doing a software autofocus will really increase the total imaging time. Another problem is that if your image or your sample has a low contrast or dim signal, it's not going to work because this technique is based on the contrast. And another problem is that if you have a sample that is slightly bigger than just flat, So for example, if you have a tissue that is a little bit thicker than just one flat cell, or if you have vesicles in the cell, I mean, basically, anytime you're using a high NA objective anyway, that doesn't work, because you can imagine that there will be at each Z plane, you will have some things that are in good contrast, and therefore the software will not know which level to choose, which Z level to choose. So it only works if there is only one possible focus, basically. The advantages is that it's available on upright and inverted systems, and that it's available on pretty much all the microscopes that have automated Z. Now we have a second solution. Instead of software autofocus, we have hardware autofocus. And again, all these have different names. Sometimes we can call this laser autofocus, but it's the same thing. So how does that work? So let's say that we have this cell on the cover slip, right? And we would like to focus on the nucleus. So there is a distance between the top of the cover slip and the plane that we would like to image, but it's always the same distance. So let's say we have 3 microns between the top of the cover slip and the plane of interest. So we are, what we do is that we shine some light with a very long wavelength, so it doesn't interfere with the sample. So it's like, it's over 750, 770. So it's like usually 870 or something nanometers. So very long wavelength light. Is sent into the objective, and then there is refraction on the interface, because here we have glass, and here we have something that has the water refraction index, so we have refraction. Some of this very long wavelength light is going to go back into the objective, and then it goes to a little detector, which will detect the interface. So basically, the machine then knows where the interface is, and you can put an offset and you say, well, I want to go 3 microns above the interface. So if there is a focus drift and the sample goes down, for example, the software would detect that the interface and the distance between the focal plane and the interface has changed, and it will correct and move an extra focus length to go back to the correct focus. So using this, you are able to continuously in the background without you even knowing it. So it doesn't take any time. It's just running in the background. You can correct the focus and keep the same focus without bleaching the sample, because it's a very long wavelength. So there are disadvantages. First, this hardware autofocus requires a refraction index interface. It will not work if we have a sample that is mounted in, for example, prolonged glass, so that has a very high refraction index, the same refraction index as glass. And then we have the cover slip, and then we have an oil objective. So all this has the same refraction index, and therefore there will be no refraction. So this system is based on having refraction, so it only works for imperfect setup. Basically, if your setup is perfect and everything is matched, you will not be able to use it because there is no refraction index interface. And then it doesn't work if the focus of interest changes. So if you have a sample where you have, for example, if you have a gel and you have some cells on top of the gel and the gel is not flat, sometimes the distance between the refraction index interface and the plane that you would like to image is different from one area in the sample to another. So this only works if you always have the same distance between the refraction index interface and the plane of interest. And it's not available on upright systems. So this is not something we can use. But there are some advantages. It is instantaneous. It runs in the background. You do not even know that it's happening. So it doesn't take any time from your experiment. There is no bleaching of your sample because there's no imaging. And any application where the sample is at a fixed distance from the cover slip can benefit from a hardware autofocus. So the names for the main companies, Nikon calls it perfect focus system, which is I hate because it's abbreviated PFS, which you will understand confuses me like hell compared to the point spread function, which is PSF. So I mix them up all the time. Anyway, perfect focus system. And then Zeiss calls it definite focus. Leica calls it AFC, Adaptive Focus Control, and Olympus calls it ZDC, Zero Drift Control. So one of the assignments that you have now is that you're going to have to go back to your microscope and figure out if you have a hardware autofocus on your system. So at least you know the names, so you can ask the person in charge and then ask them to show you how to use it. So this is the way the Nikon perfect focus system works. We have this LED, which is a very long wavelength, and you can see here the dichroic mirror. So it's around 800 something, 870, I think, that the light goes through. So everything below, so this is a multi-band pass filter, right? And and so everything that is below, which is what we're imaging, will be transmitted, but the laser light at this very long wavelength is going to be reflected. And so here we have the laser light, and here we have some mirrors to direct the light. We have an offset lens, and this goes to the dichroic mirror, and so the laser light is reflected. Whatever is refracted at the interface goes back down, and it goes back into the autofocus system, and it reaches a sensor, and depending on where the interface has been found, then there is an offset lens that moves, and this is the place where we're imaging, so it doesn't interfere with imaging. Now, these techniques do not work if we have slanted or complex surfaces that are not regular. So for example, if we have a gel and we seed cells on top of the gel, and the gel always retracts in the center, and it is higher on the edges of the dish. So what we can do there is use something with the software where we're going to make a complex software surface to image. So we can go to this first point here of the gel and focus and record the first X, Y, and Z. And then we go to another place in the middle, and the objective is lower because the sample is lower. And we're going to again record X, Y, Z, and etc. So we do this with many points. And once we think that we have a good sampled very well the whole surface, then the software is going to create an imaging surface, which is lower at the center, and it interpolates everything. And so that means that you can go to places that you have not measured and be in focus. So the disadvantage is that the positions need to be entered manually, at least the reference positions. The more complex your surface and the more positions you need to be representative. But the advantages is that it works for uneven surfaces, like sloped surfaces, for example. And it also works if you have no refraction index interface. And it works if the sample is not flat. So this is also a good strategy that you should have in probably have in your microscope. If you have a high-end microscope, you have this sort of software, so you can go and ask as well. So this is a little conclusion here. And if you use the automation of the you should use the automation of the microscope to explore your sample and find the area of interest. You will have less bleaching, you will use less time, and less risks to image twice the same area. So take an overview of your sample and use the image, the overview image, to navigate in your sample and record the positions of interest. You need a reliable focus, and this is critical to get reliable image analysis. So you need a focus strategy, which is in two parts. Number one, how are you going to focus to start with, so that next time, the next day, when you focus on the next sample, you actually focus at the same type of place. And number two, how are you going to keep the focus while you're imaging? So what will you focus on to ensure that images of different samples are comparable? If you are using a sample that is on the cover slip, if these are cells, for example, filopodia are very thin. So it's a very good way to have the sample always at the same focus, always start at the same place if you focus on the filopodia. But this is very variable from one sample to another. Another way to have a strategy to start is if you're imaging something inside the sample, you could find the equator of the nucleus, for example, so the widest place of the nucleus. That could be also a good way to start for your imaging. So you have to figure out how you're going to start focusing so that the next day you focus at the same place. And then you need to find a structure that is not affected by your treatment and mutation. Clearly, if I don't know, if the filopodia disappear when you are doing a mutation, you're not going to choose the filopodia to make sure that you always image at the same plane. And then you need to write your focus strategy in material methods in article, because it's important for the reproducibility of the method. The next person who wants to reproduce your method should know exactly what your focus strategy was, and then try to use a hardware autofocus whenever it's possible. That's it for me. This is for Monday.